Hello, and welcome back to the second half of this lecture where we're taking a look at DNA analysis. And so in the first half, we took a look at a way in which we can go through and find the exact sequence of the A's, the T's, the G's, and the C's, the exact sequence of the information that's coded within the DNA molecule. In this half of the lecture, we're going to take a look at genetic testing. So basically, how is it that we're able to use this information in a way in a human medical setting? So basically what we're going to be doing is similar to what we've talked about before. Genetic testing is going to go through and it's going to identify the alleles of an individual's genes. And so it's basically going to tell us whether or not an individual has kind of what would be referred to as kind of a normal version of the gene, or if they've got an altered version of the gene. And if they've got an altered version of the gene, it just means that that DNA sequence is a little bit different from what's normally found within the population. And because it's a little bit different, what could happen is that it could lead to a predisposition or lead to causing some type of disease. Because many of the diseases, we take a look at it, are inherited genetically. And so in this case, what we've got over here is basically the idea that we've got an unaffected father, an unaffected mother. Each one is a carrier for one of those recessive genes. Basically, it's a gene that doesn't show up unless you've got two copies of it, two alleles, have the kind of altered version of that information. So as long as you've got one copy of the normal one, you're fine. And if you put that through, and this is using that kind of square type orientation that we talked about in one of the previous lectures, don't worry about crossing this up, but this basically represents the different types of eggs from the mother, sperm from the father, that could come together and basically end up with a situation that, you know, one person is completely unaffected, 1 out of 4 offspring. In theory, if you had 4 offspring, kind of randomly, this would occur. 2 out of 4 would be unaffected, but would be carriers. So they'd have a similar composition of their DNA to their parents. And then finally, we've got this person over here that received kind of that altered allele, that altered information from the mother, and they received that altered allele, that altered information from their father. So in this case, they've got both copies of that genetic information, are the altered, affected, kind of changed region of that DNA. So this individual, on an average about 1 out of 4 times, is going to be affected by the disease. And therefore, they're going to develop the disease, or they have the potential to develop the disease. And they're likely going to pass that trait on to their offspring. In this case, the 2 unaffected carriers are going to have a risk of transmitting that information, transmitting that altered gene to their offspring as well. And so what we can see is that in the field of genetic counseling, we have the ability to kind of study and determine what are your risks. If you're a carrier, what are your risks of having a child with this genetic disease or this genetic disorder? And the only way we can do that, previously we'd have to do it by, heredity charts and pedigrees and things like that. Now we can go through and study the DNA and get a little bit better understanding about what's going on. So if we're going to do this, what you can see is in a laboratory setting, normally what's going to happen is we're going to collect cells that are going to have the DNA. So the easiest way to do that is you go through, it's called a buccal smear, which is, again, a nice, fancy, overly complicated way of saying that we're going to go in, we're going to use a scientific Q-tip, which means it's sterile and designed to collect these cells. We're going to scrape it along the inside cheek of the mouth. And when we do that, this is going to collect these cells, and the cells are going to have the DNA in them. And so they're going to be on this tissue, this little swab, and we're going to put it in a tube, and we're going to study that DNA. We could do the same thing by taking a little bit of blood from an infant and studying the DNA that are in the nucleated cells within the bud. If you're doing this at an at-home test, like the 23andMe or I can't remember the other one, Ancestry.com, I think is the other one, the 2 big ones. You basically, spit into a test tube, so your saliva is going to contain some of these cells, and those cells are going to contain the DNA. And so basically, you get it, whether you're doing it with a tissue sample here, with a swab, blood cells from an infant, or saliva. If you're doing the at-home test, you're going to get the DNA, and it's going to go through that process of sequencing that we talked about before. Now, we can also do this as a prenatal diagnosis, and this has been a process we've been doing for a number of years for looking at chromosomal abnormalities and sex determination, because you can go through, take a look at the cells and say, okay, do they have, the right, 22 pairs of autosomes and an X and an X or an X and a Y. So you can go through and see if they've got, missing chromosomes, if they got duplicated chromosomes in some way, or triplicate chromosomes. We don't, Down syndrome is the example we used, was trisomy 21, 3 copies of the chromosome instead of number 2, number 2 copies. Sex determination, take a look at, do you have 2 X chromosomes or X and Y? But now we can go through and actually take the DNA and study the genetics itself. In the process of amniocentesis, what we do is basically use kind of a specialized syringe, insert it into the mother's abdomen using an ultrasound, kind of track the way this needle goes in, and collect some of the amniotic fluid. The amniotic fluid is that fluid-filled sac that kind of wraps around the fetus within the placenta and cushions it. But the important thing is that within the amniotic fluid is going to be a collection of fetal cells. And so we can collect these cells and study the DNA from them, similar to the way that we would do it in the situation here. So genetic testing, normally done with a genetic counselor, is recommended if a woman is pregnant over the age of 34, if there was a prenatal screening test that had, some type of abnormality associated with it. If the couple plans to have a family and one of them or a close relative has an inherited illness, and they're concerned about knowing what are the risk factors of passing that on to their new child, or if the child already has serious birth defects or serious genetic disorder, what are the odds that, they're going to have another child that has that disorder? Again, not making a decision as to what to do with that information, but using the science and the technology to provide information to the prospective parents. Now, genetic counselors are going to be specialized, trained clinicians. So they're like specialized nurses, specialized counselors that understand the genetics and understand how to work with people in very trying and emotional situations. But they basically sit down and go over the results with the people that are going through that genetic testing and explain it to them. Because, the cold hard fact is that, if you get an ancestry.com kind of readout, you have no idea what it means. You're going to go on, internet, check with Dr. Google and find out what it all means. Get some information that may or may not be accurate. And so the genetic counselors there are helping you to understand what that genetic information means, what it implies, and help you kind of think about how it's going to impact you and potentially what decisions you'd want to make. Now, again, this is talking about the science. The science says we can do this. The science says we can understand, what is the risk of having an offspring with this genetic disorder? What is the risk if we, do an amniocentesis and study it? What can we learn about this infant before it's born? But there are risks associated with it. One example of this, China in the late 1970s launched a program to slow their population growth. And so they basically had a one-child policy. Previously, they had, kind of uncontrolled population growth. They were growing kind of beyond their resources. And so they were, seeing a lot of poverty associated with it. And so basically they said that, each family can have one child instead of kind of replacing themselves in the next generation. They're actually going to have, 2 individuals, a mother and a father, coming together to have one offspring, to have one child. And that would be, slowing the growth of the population, potentially causing the population to decline in some areas. Now they had some caveats with this. They had some exceptions with this in rural areas, in history. They're often dependent upon the family working on a farm or contributing to the family income. And so in rural areas, many people were allowed to have a second child if the first child was a girl. Again, a fairly sexist view. The women wouldn't be working in the field or working in the family, business or something like that. But culturally, there was this idea that women would be involved with maintaining the homestead, raising the family, and the boys would grow up to be men that would be the providers and the leaders of their families. And so, because of that, with that one-child rule policy or one-child policy. They basically, in that society at that period of time, began to see a preference for having boys. And so they would go through this testing, in many cases, abort the child if it was a girl because they had a preference for it being female. What happened over a period of time, and so if we take a look at this, the fertility rate through the 60s, through about 1980 when the policy went into effect, and beyond, what they saw is that most women were having 6 or 7 children prior to the 1970s. It had already started to come down. They implemented the one-child policy about 1980, and then stabilized it at about 1.5 children per female. And so it potentially had the effect that they wanted. It wasn't this huge population growth that they saw previously, but that birth rate was already declining. But it's stayed relatively stable for a period of 10 or 15 years. This is, again, it's the most recent information I was able to find on this. But what did change, because they had a male preference, is that there was a ratio to about 120 boys being born for every 100 girls. And so what this means then is if we take a look at the sex ratio of males to females, we started to see many, many more males being born as opposed to females. Normally, it's going to be pretty close to 1-to-1, a little bit different, a little bit of variability, but this was changing within China. And this resulted in about 30 million surplus boys being born. So 30 million boys, 30 million more boys than girls born during this period of time. And so this policy was put in about 1980, was relaxed at about 2015, allowing people to have 2 children and this was done to address the sex ratio, to bring that closer to that 1-to-1 ratio between boys and girls, as well as to respond to a population that was aging, and they were running the risk of having a younger generation that wouldn't be able to support the needs of that aging population. Another example, not looking at sex, taking a look at males versus females, but looking at genetic disorders. Things that can be tested for now are things like Tay-Sachs disease. Tay-Sachs is a progressive destruction disease, genetically inherited, that results in degradation and destruction of the nervous system and brain. It's generally lethal by the time the child reaches about age 5. Another example is cystic fibrosis, a good, simple genetic disorder. We'll talk about this cystic fibrosis as an example later on. Because it is a single gene, it's something that could be potentially affected if we could repair that DNA. And we'll talk about that with genetic recombination later on. But basically what happens is it has a defective protein within the lungs, causes mucus to build up, and that mucus then makes it harder to breathe, makes them susceptible to infection, and because of that, it's generally lethal by the time the individuals reach early to mid-adulthood. Now again, medicine has gotten better, so individuals are living a little bit longer, but it still continues to be the most common fatal genetic disease that we're facing now at this point. And again, this information can be learned by studying the DNA of the individual and informing the parents. For some individuals, they may decide to, abort the child so that, the child doesn't suffer through these things. Others will, try to, have the child and make the best life for them. But again, this is science. The science says we can sequence the DNA, we can identify whether or not the child is at risk for Tay-Sachs or cystic fibrosis, but it doesn't tell you what to do with that information. That's where the ethics comes in. And so the readings for this half of the lecture is talking a little bit more about these concepts of genetic testing, Help me understand genetic testing from the National Institutes of Health, National Library of Medicine. Help me understand, actually I've got this in there twice. One is the testing process. One is actually the direct-to-consumer genetic testing. And then I also have an article for you to take a look at the direct-to-consumer tests as it's described by the U.S. Food and Drug Administration. And so the one up here is like the genetic testing that you would get if you went to a doctor and saw a clinician, saw a genetic counselor. The ones down here, the direct-to-consumers, are the 23andMe or the Ancestry kits, where you spit in a test tube and a couple weeks later they send you your DNA results without a whole lot of explanation, and you've got to try to interpret what it is you're doing. There are a lot of concerns about individuals having that information and not knowing what to do with it, which is why they really encourage people that have genetic disorders or fear of having children with genetic disorders that they go through a process of genetic counseling. As always, if you have any questions, feel free to email me. Thank you and have a great day.